Hello, my name is Kyle Parker, and today I'm going to discuss MREB3018, which is a gene in MRIBR, as an ortholog to E.coli CasB B2759, which is a protein that functions in CRISPR's E. coli defense system. Now, what is Myothermus rubra? Myothermus rubra, or MRIBR, is a bacteria discovered by Loganova and Ogorova in a Russian hot spring. The characteristics of it are that it's gram-negative bacteria, it is rod-shaped, it's red in color, and it grows in temperatures 35 degrees Celsius to 70 degrees Celsius with an optimal temperature of 60 degrees Celsius. Now, why study MRIBR? MRIBR only has around 100 articles posted on it in its lifetime, and its family of bacteria also only has very few articles posted on it in its lifetime. Studying extremophiles may find us that it has evolved to function differently and may change the way we think about things like CRISPR, especially in E. coli, which is why we are going to compare it to E. coli as the model system. CRISPR in E. coli is a part of a bacteria defense system against bacteriophages. E. coli has a Cas1e type CRISPR, which functions in 3 steps. The first step is spacer acquisition. In spacer acquisition, the cell recognizes a foreign PAM sequence, which is a protospacer adjacent motif. The protospacer adjacent motif, or I guess the protospacer, is a part of the bacteriophage's invading DNA that will be incorporated into pre-CR-RNA that will be used by the CRISPR system to detect future invading bacteriophages. The way that we obtain this protospacer is by first, the cell recognizes the PAM sequence. It will then recruit Cas1 and Cas2, which are proteins in the CRISPR system, to go and hold the invading bacteriophage DNA. They will then recruit Cas3, another CRISPR system protein, which will then cut out the protospacer. It will be then turned into a spacer and inserted into pre-CR RNA. In the next step, CRISPR-Cas expression or CR-RNA biogenesis, the pre-CR RNA is turned into mature CR RNA. The mature CR RNA has 3 parts: the leader sequence, the repeats, and the spacer. Shown below in the bottom right in the picture, with not pictured first, is the leader sequence, which contains the promoter for the CR RNA to work, and it's also the part that is read first in the CR RNA. Next is the repeats, which are palindromic and can form hairpins. They occur before and after all spacers. Next is the spacer. The spacer is what has been acquired in the last step from the foreign bacteriophage DNA and will be used in the next step to fight off future bacteriophages that it recognizes. In the DNA interference step, once the cell recognizes a foreign bacteriophage DNA that it has a spacer from, the spacer will bind to the protospacer it recognizes and form an R loop. The R loop will then be recognized by the CRISPR system, which will then recruit Cas-A, another protein that is used. And then once Cas-A is bound to the R loop, Cas-A will recruit Cas3. Again, Cas3 will then degrade the DNA, which will then interrupt the bacteriophage from doing what it usually does when it invades bacteria. This is the CRISPR system and is also called the CRISPR cascade and how it works. The other proteins that were not mentioned are CasB, CasC, CasD, and CasE are part of a structural and form a more structural formation of an S. This S, which is headed by CasE and tailed by CasD, is held together by CasB and CasC, which will form the backbone and is held in the front and the back by CasB. CasB will then bind the DNA to itself when it notices the bacteriophage DNA so that Cas2, Cas1, and Cas3 can cut off the spacer or degrade the DNA. CasB is important for CRISPR function because if it is removed from the CRISPR system no longer functions. Now, in order to determine that MREB3018 is CasB in E. coli's B2759 gene and that they are orthologs, we use free bioinformatic tools that are recommended by our professor, Dr. Scott, the first of which is KEGG. KEGG, when it analyzed MREB3018, showed the product CasB. It gave an amino acid number for MREB3018 of 202, and it gave the coordinates of 305, 7916 to 305, 8524. When it analyzed E. coli, it gave the product of CasB again, an amino acid number of 160, and the coordinates of 288, 2155 to 288, 2637. While it did give a different amino acid number at different coordinates, it produced the same product, which is important because evolutionary differences could have changed the amino acid number and coordinates. Next row shows NCBI BLAST data done from a pairwise sequence alignment between MREB3018 and B2759. The E value was 1.5 and the percent identity was 50. This indicates that the sequences were most likely matched up by chance and are not similar in a pairwise sequence alignment. This is most likely due to evolutionary differences and a different amino acid number. Next was COG. COG will analyze the sequence and then look through its database to find protein domains that are similar to the protein that it has analyzed. In this case, it gave a different COG number, but the same COG name, which is CSE2-1E, which is a different name for CasB. Also in this case, the score was very high and the E value was very low, indicating that these two proteins are part of the same domain. The next three rows are about cellular location. P sort B will give a score to each part of the cell and decide where the protein functions in the cell. For MREB3018, it gave the highest score to cytoplasm, indicating it is part of the cytoplasm. In E. coli B2759, it gave the location of unknown because it gave the same score to each part of the cell. The next row is TM-HMM, which will analyze the protein sequence and then decide if the protein has any transmembrane helices. In both MREB and E. coli, no transmembrane helices were present, so it is not part of the membrane. In PRED, which is the next row, PRED will analyze the protein sequence and then decide if it has any beta barrels present. When it analyzed the sequence, no beta barrels were present for MREB or E. coli, also indicating that it is not part of the membrane. In E. coli, since it is not part of the membrane and using background information, it is most likely to conclude that the protein functions in the cytoplasm. The next row is TigerFam. TigerFam will analyze the protein sequence and then decide what protein family the analyzed sequence is a part of. For both MREB3018 and E. coli B2759, the TigerFam number and name was the same. To confirm this, the score was 118 and 177.8, which is very high, and the E values were also low, indicating that the sequences are not matched by random chance, and that these two proteins are part of the same family. The next row is PFAM, which also give the same PFAM number and name, but instead of indicating which protein family it is a part of, it will indicate protein domain, like COG. The E value was also very low in this case, indicating that these two proteins are both CRISPR-Cas9 and they are both part of the same domain. Next is PDB. PDB will give a crystallized picture of your protein after it has analyzed your protein sequence, which are shown at the bottom, and it will also give a protein family name. For MREB, it gave CSE2, which is also CasB, and for PDB, it gave the cascade. Now, the cascade is the entire CRISPR system, but it did put an emphasis on CasB, which indicates that these are part of the same protein family. Now, before we did the pairwise sequence alignment, we wanted to determine that the start codon was in the correct place. We did this using IMG. IMG gave us a picture of the chromosome 30 amino acids before the start codon that was predicted, and showed the Shindell-Garno sequence if that was correct. The Shindell-Garno sequence is in light blue and green, and the start codon it predicted is in yellow and red. The yellow and red is the only start codon that can be predicted, despite the fact that it is GTG instead of ATG as a regular start codon. However, it does encode an M, which means that it is the correct start codon. No other start codon was found close enough to a Shindell-Garno sequence, so this confirmed that this is the correct start codon. We then did an NCBI BLAST multiple sequence alignment to confirm with other bacteria similar to M. rubra that their start codon was in the same place, and on the bottom you can see that the M's are all lined up, indicating their start codon was also in the same place. After this, we did an NCBI BLAST pairwise sequence alignment between MREB 3018 and E. coli B2759, which, as mentioned above, had an E-value of 1.5 and bit score of 14.2, indicating they are matched up by random chance and are not the same sequence. This is most likely due to evolutionary changes and a different amino acid number. Next, we did a cellular location study using TM-HMM at the top, PRED on the bottom left, and P-B on the bottom right. TM-HMM at the top, which shows MREB on the left and E. coli on the right. If a spike was present in the graph, then a transmembrane helice would be present, but since no spikes are present, no transmembrane helices are present for either of these proteins, indicating that they are not part of the membrane. PRED will show if beta barrel is present if consistent spikes are present, but consistent spikes are not present, indicating that this is not a beta barrel for either protein, either MREB on the left or E. coli on the right. The final is P-B on the bottom right table, showing the highest score for cytoplasm, indicating MREB 3018 is part of the cytoplasm, and E. coli shows the same score for each one, but using previous cellular location tools, it is most likely part of the cytoplasm as well. Now, to determine if MREB 3018 is part of an operon like E. coli is, we use KEGG and IMG. On the left, KEGG showed a picture of the chromosome near our gene of interest, and if it is flanked by genes that are part of the CRISPR system, then is part of an operon. It is flanked by MREB 3017 and 3019, which are both part of the CRISPR system, indicating it is an operon. The bottom picture also shows the E. coli system, which is flanked by CasA and CasC, which are also part of the CRISPR system, indicating it is also part of an operon. To confirm that MREB is part of an operon, MREB 3018 was part of an operon, we compared it to other similar bacteria, and they were also flanked by the same MREB 3017 and 3018 or 3019 genes, indicating that MREB should be part of an operon. The final test was a paralog test, in which MREB 3018 was compared to glycotransferase. The E-value was 7.3 and the bit score was 23.5, indicating they were matched up by a random chance, and E. coli or MREB 3018 does not have a paralog. Given that the bioinformatics data presented above, it is predicted that MREB 3018 is orthologous to E. coli B2759 CasB. The hits when pulled from PFAM and COG indicate the proteins are in the same domain. The hits from TigerFAM and PDB indicate that the proteins are in the same family. They also have the same cellular location, they're both part of an operon, and they produce the same product, indicating that they are orthologous. I hope this information was informative. Thank you for your time.